All right, it's coming through on Twitter.
Okay, where's the record button? There we go. Okay.
And we're coming through on YouTube as well.
Okay. Just do one more thing and then we'll be ready to go. Okay. All right. I will go ahead and get started, and if people jump in afterwards, that's fine as well. So I am going to do something a little bit different today than we would normally see during these graduate student seminars. So normally it would be a graduate student who is presenting what they've been working on during their time as a graduate student. But considering that I just did that last spring, and that fingers crossed I will be defending this upcoming spring, I decided to give you a break from what I'm doing and actually highlight the work that all of the students in Dr. Armstrong's research group are doing. And so collectively, we call ourselves the Soil Ecosystem and Nutrient Dynamics Lab. And so today I'll be talking about cover cropping in the Midwest, an overview of our research program. And so I just wanted to start with kind of how we approach our research program. And so we're pretty focused on the evaluation and development of conservation practices in row crop agriculture. Along the left there, you can see all of the different areas that we work in. So some of the big ones are agronomics, nutrient cycling, the soil microbiome, water quality, remote sensing, and then we also are getting into some of the economics. And for us, our job isn't complete until we transfer the knowledge from our research to either the scientific community or the farming community. And so we do that through publications, student development, field days, training and workshops for CCAs, producers, government agencies, and extension publications. And so when we tend to think about when we approach a research question, we tend to approach it from the point of view of producers. And so when we think about producers who use cover crops in their system, we try to think about, as they're going through the field planting their corn in the spring, what kind of questions are running through their head about the cover crop system. And so some of those questions are, how much biomass did I generate? How much nitrogen is in that biomass? How much of that nitrogen will my crop actually be able to use? And at what point in the year will it be available? How much carbon did I generate? How is the soil microbiome responding to that addition of carbon from the cover crop residue? Should I expect any nitrogen immobilization, and do I need to adjust my fertilizer management to account for it? Do I need starter nitrogen? And if I do need starter nitrogen, how much do I need? And then lastly, how do I maintain my yields, right? And this is really one of the biggest ones because the yields are the producer's livelihood, right? And so we need to be able to promote this practice while also ensuring them that they're not going to see a huge hit in their pocketbooks, right? And so what I'm going to do today is run through some of our research projects that have given us a beginning to the answers of to these questions. And so what I want to start with is actually the first big project that we really had as a group that began back in 2014. And so this is actually a combination of two projects that were both funded by the Illinois Nutrient Research and Education Council. And what we did in these is we're looking at the effect of cover crops on nutrient loading through subsurface drainage at multiple scales. And so we'll start by looking at the field scale. And so back in 2014, we set up this research location with 15 individually tile drained plots. And so you can see the tile drainage lines in the red coming to the yellow squares, which represents the tile monitoring stations, which you can see in the picture on the right-hand side. And so these allow us to actually tap into the tile lines, pull samples from water during storm events, and analyze them for nitrogen and phosphorus content. Today, we'll be focusing just on the nitrogen side of things. And so the first thing that we need to look at is how well did our cover crops perform in these systems when we, when we think about what's the effect of the cover crop versus a non-cover crop system. If you plant a cover crop and don't have any production from it, then I wouldn't be surprised to see no effect, right? So we need to make sure that we're having at least a representative cover crop stand comparable to what producers would typically have. And so over 5 years, we had an average of about 1,300 kilograms per hectare of above ground biomass with a nitrogen content in the above ground biomass of about 43 kilograms of nitrogen per hectare and a total carbon accumulation of about 520 kilograms per hectare. And so one important thing to note is that we typically terminate our cover crops prior to stem elongation. And so at this point in time, they have a lower C to N ratio than they would had the crop matured longer. And so on average, we had a shoot C to carbon to nitrogen ratio of about 12:1. And so this nitrogen uptake by the cover crop actually leads to a lower amount of nitrogen within the soil profile, which gave us hope that we could actually reduce the tile or the nitrate in the tile drainage. And what we actually found was that over 5 years, the cumulative nitrate load between cover crop and non-cover crop systems, the cover crop had reduced nitrate loading by about 47%. And so this is interesting because in many of the state nutrient reduction strategies that were developed to help mitigate the water quality issues leading to the Gulf of Mexico hypoxic zone, they're calling for about a 30% reduction in nitrate loading from cover crop fields. And so currently at this point in time, we're actually outperforming the estimated reduction that we would need from cover crop systems. And so this is great, but this is all at a field scale. And so once we saw this, we had a question that we wanted to answer, and that was, will this effect of cover crops that we see at the field scale scale up and actually be reproducible at the watershed scale? And so we have a project, the assessment of cover crops at the watershed scale, again, funded by Illinois Nutrient Research and Education Council. And what we have in this project is two sub-watersheds within the Lake Bloomington watershed, just outside of Towanda, Illinois. We have a control watershed that has no cover crops, that's about 260 hectares, and a cover crop watershed, or the treatment watershed, that is about 465 hectares. And so within the cover crop watershed, about 50% of the land area is actually planted to cover crops. And so the 93% of all the land that is in these watersheds is row crop agriculture. So there's not really any influence of urban runoff or anything like that. And we actually were able to involve 10 farmer collaborators in this project. And so here's just an image of the cover crops being flown on. So one thing that I want to point out between the field scale project and the watershed scale project is that in the field scale research, it's very highly controlled, right? So all of the management across all of the treatments is the exact same. We knew going in what cover crop we were going to use. We knew what year was going to be corn and what year was going to be soybeans. And so the biggest difference is that in the watershed, we didn't control any of that. So we let the producers, do whatever their typical nitrogen management practices would be. Any changes that they saw that they needed because of the cover crops, we let them choose what species they were going to plant. Now, interestingly enough, they all kind of got together and came to basically a collective decision amongst themselves that they wanted to go cereal rye radish before soybeans and oats radish before corn, but we didn't tell them that they needed to do that. And then the other thing is that, obviously they've been planting these fields for years. And so it was a continuation of their normal rotation. We didn't try to change anything for them. The only thing that we do is pay for the cover crop to get flown on, sample the cover crops, and sample the water that's leaving the watershed outlet. And so, it's a very farmer-driven type of research project. They had a lot of control, and that what we found though is that in fact the results that we did see at the field scale actually did translate to the watershed scale, and this is with only 50% of the watershed being cover cropped. And so you can see here that over this 4-year time period that I'm showing, we had about a 33 to 36% reduction in nitrate loss via the tile lines. And so what this is really telling us is that we can achieve these water quality results that we're looking for at the watershed scale without actually having to cover crop the entire watershed. And so maybe we need to target certain producers who are in high risk areas for nutrient loss. And if we can get, a certain percentage of the watershed covered, then we may be able to achieve the results that we're looking for. And so this is great. And we can, we're happy about the environmental benefits that we're seeing from cover crops. But what remains though is a really big question concerning the impact of cover crops, and specifically cereal rye, on cash crop yields. And so again, going back to that field scale project that I showed at the beginning, when we look at the effect on soybean yield across the 2 years where we had soybeans at the site, we saw no significant difference in soybean yields. And in fact, this is common in the literature. Typically, there is no effect reported, and sometimes even positive effects. And so this is kind of what we expected to see. But when we look at the results for corn yields, we saw a 13% reduction in corn yields over a 3-year period for corn following cereal rye compared to the control. So this isn't great. This is actually higher than the typical yield reductions that are reported in literature, which is about 6%. But what I'll say is that this result is not unique. And so part of my research is looking across the entire upper Mississippi River Basin. And what I found when looking at 430 paired site years for corn yields, I saw that on average, there was a 6% reduction in yield following cereal rye compared to the control. And that was regardless of any fertilizer management, length of cover cropping, tenure, any of that. And so this kind of reduction that we're seeing isn't unique, but it is important that we dig further into it. And so one thing that we did was we sampled at different growth stages for nitrogen uptake within the corn. And this is a 3-year average. And what we have seen is that by the time corn reaches physiological maturity, so that R6 growth stage, there's actually about a 28% reduction in the total nitrogen within the plant for corn following cereal rye compared to the control corn. The other thing that I want to point out here is that this nitrogen gap actually starts to form about 60 to 75 days after cereal rye termination. And you might look at this graph and think, oh, that could be sampling error. That could be, error and just human error, right? But what I'll say is just keep this graph in mind as we move through, and I'll try to prove to you that in fact it's not sampling error, but there is actually something going on here. And so after we saw this nitrogen gap, this really got us questioning, do we need to change our fertilizer practices to maybe overcome this gap? And so one of the things that we were interested in was the use of starter nitrogen fertilizer. So Houston Miller, who just graduated last spring with his master's degree, looked into the optimization of starter nitrogen fertilizer for corn following cereal rye. And so this was a project funded by the Indiana Corn Marketing Council. Basically, we were looking at what is the best rate of starter nitrogen for corn following cereal rye to achieve competitive yields to the control. And so the first thing that we did was we did that same sequential growth stage sampling of corn and looked at nitrogen uptake. And what we found was that we were actually able to, so first of all, I want to point out, you can see the gap beginning to form right here around this, V11, V12 growth stage. You can see it beginning to form. And so again, this just kind of shows that that gap that earlier was not an artifact of sampling. There is actually something physiologically going on here. What we don't quite know the answer to that yet, but we are looking into it to see if we can overcome what's happening there. And so what we found was actually that we were able to reduce the nitrogen gap from 13%, which is between the control and the cereal rye without starter, to it was about 4%, a 4% gap in nitrogen uptake when we had 84 kilograms of nitrogen as starter fertilizer. Okay, so we were able to overcome some of that nitrogen gap. There's still a little bit there, but we were able to overcome some of it. And so now let's look at how we were able to affect yields using starter nitrogen. And so what we found was that at only 1 out of 3 of the locations where this experiment took place did cereal rye even significantly reduce corn yield. But at 2 of the 3 sites, when we added 56 kilograms of nitrogen per hectare, we were actually able to achieve greater corn yield relative to the non-cereal rye with no starter control. And at all 3 sites, the addition of 28 to 56 kilograms of nitrogen as starter resulted in greater yield for corn following cereal rye compared to the corn following cereal rye where we had 0 starter. And here on the graph on the left, you can see that the yields were relatively equal. And so, okay, so we're starting to overcome some of the problems. And so one of the questions that we get all the time when we present to producers as well as other groups is, okay, so all of that nitrogen is in the cereal rye biomass. How much of that nitrogen is my corn actually going to be able to use? And so this is where Corey Lacey's research comes in. And this is work that was funded by Indiana Corn Marketing Council, Indiana Soybean Alliance, as well as Illinois Nutrient Research and Education Council. And basically what he did was use 15N stable isotope techniques to track nitrogen from the cereal rye biomass into the corn and soybean crop. And so basically we grew the cereal rye, pulse labeling it with 15N labeled ammonium nitrate. We then harvested the cereal rye residue and placed it onto these microplots, which you can see in the picture here, where we would eventually plant the corn and soybeans at. So following planting, so this is the example for corn, we sampled the plants at V6, R1, and physiological maturity, and then used isotope ratio mass spectrometry to determine the amount of nitrogen in the corn or soybeans that actually came from the cereal rye. And what we found was that by R6 or harvest time, about 10.5% of the cereal rye nitrogen had made it into the corn, and about 12% of the cereal rye nitrogen had made it into the soybeans. And so then what we wanted to do was look at how the nitrogen within the total plant was partitioned between the grain and the stover. And so what we found was that in the corn, about 9% ended up in the grain, with a 1.5% in the stover, and in soybeans, about 8% in the grain and 4% in the stover. But the really important thing to take away from these graphs is that 90% there that is in neither the corn or the grain or the stover. And so what this means is that nitrogen is either A, in undecomposed residue, so it has yet to break down and release, or B, it's in the soil profile. And so seeing these results kind of led us to question, well, you know, if it's not ending up in the cash crop, where is it actually going when it releases? Where is the nitrogen going when it releases? And so I wrote a North Central SARE graduate student grant to actually answer this question. And so I used the same 15N isotope techniques to look at where within the soil profile did the nitrogen from the shoot and root portion of the cereal rye plant actually end up. And so what I did was I used an incubation method to determine this. And so I added to the incubation microcosms, I added either labeled or non-labeled shoot and root residue and incubated across a simulated corn growing season. And so I simulated the number of growing degree days that would accumulate between cereal rye termination and corn physiological maturity, and sampled sequentially through there at what would be representative of key growth stages for corn. And during each sampling day, I analyzed for microbial biomass nitrogen, inorganic nitrogen, as well as total nitrogen. And then again, I used that same isotope ratio mass spectrometry technique to determine the portion from cereal rye that was actually in each nitrogen pool in the soil. And so basically what I found was that over the course of that simulated growing season, 67% of the nitrogen contained within the whole plant, so meaning shoots and roots, was released to the soil in some form. And so what I'll say is that of that 67%, the majority of that is from the shoot portion of the plant. And so over the course of this time period that I'm showing here, if we were looking at just shoots, we saw about 90% of the nitrogen in the shoot portion of the plant be released. And much less in the root portion. And this is simply because the root portion of the plant has a much higher C to N ratio. And so it's much slower to actually decompose and release the nitrogen. And so the next thing that we wanted to answer is, where does this nitrogen end up within the soil? And so I looked at that as well. And if you focus on the bar on the far right, the white with black dots is undecomposed cereal rye nitrogen. So this is nitrogen that's still in the residue. Black would be microbial biomass nitrogen. Green is inorganic nitrogen, and gray is nitrogen in the organic form. And what we found is that at the end of that simulated period that I had, 53% of the cereal rye nitrogen was in the organic form within the soil, 15% was in the inorganic form, less than 1% was within the microbial biomass, and 32% remained undecomposed. And so another interesting point here is that the majority of the nitrogen release, especially the inorganic nitrogen form, occurred later in the growing season, past about V12, of what would be V12 in the field. And so really the takeaway here is that we can now confirm that we should not be counting on cereal rye for any kind of nitrogen credit. And so I'm now going to switch directions completely, and I'm going to talk about some of the remote sensing work that's being done in our lab. And so this is being done by Kenru Chen, who just defended her master's thesis over the summer, is actually staying on with us as a PhD student. So that's great. We're going to get to see the continuation of this work. I have to give a thank you to NRCS for providing a lot of the data to Kenru for this project. And what she did was she evaluated remote sensing techniques for the rapid estimation of cover crops within a watershed in Indiana. And so she had several objectives, but the two that I'm going to focus on are her ability to do a hindcasting analysis of cover crop adoption within the Big Pine watershed, basically to tell us how adoption has changed over time. And then as well as she did a tenure analysis, so letting us know the length in years of cover crop use within the same fields within that watershed. And so Kenru used satellite imagery to do this research, coupled with the crop data layer and the land parcel layer to identify the different field boundaries within the watershed. And some of the results that she was able to produce from this is a look at the historical adoption of cover crops so 2014 through 2018. And you can see as we move from 2014 to 2018, there is an increase from about 5.1% of the land area planted to cover crops, all the way up to about 9%. And so this is really great to see. Unfortunately, in 2017 and 2018, the adoption seems like it may have plateaued, but we still need to get some more years onto this to see if that is the case. But you can tell here, just on the maps, the green areas represent the cover crop fields. So you can see that it is increasing primarily in the southern portion of the watershed. So the other thing that Kenru was able to do is look at cover crop tenure within the watershed. And so on the map here on the left, white would represent no cover crops, green represents cover crops for 1 year, light blue for two consecutive years, purple for 3 consecutive years, orange for 4 years, red for 5 years. And so basically, of the about 15% of the cropland that had cover crops on it at any time during the study period, about 13% of that cover cropped for 2 years or less, with only about 3% having cover cropped for 3 years or longer. And so what this tells us is that there's a lot of people trying it and potentially having a bad experience, and so they don't want to use cover crops anymore. And this research actually has a couple advantages for us, and one of them is that we can identify spots where there's very little adoption within a watershed, and actually work to target producers in that area in order to promote the practice. Okay, and so now another big thing is the economics behind cover cropping, right? So it's hard to promote a practice to producers if you can't tell them that it's going to be economical for them. And so part of my research is looking at the economic risk, benefit, and uncertainty following cereal rye adoption. And so part of our research program has been partnering with Nathan Thompson in the Ag Econ department to help us look at the economics of that field scale experiment from the very beginning of the presentation. And what we found was that there's a very high probability that the introduction of cereal rye into corn and soybean rotations actually results in negative net returns. And this is regardless of whether you receive a cost share payment or not. And so my question that I'm trying to answer is, does this trend hold true across the upper Mississippi River Basin and also as the tenure of a cover crop system increases? And so what I did was I went out and found data from across the upper Mississippi River Basin with specific criteria that the cover crop had to be cereal rye, the cash crop had to be either corn or soybeans, and each observation had to be paired, meaning that for every cereal rye plot, there was a corresponding control plot at the same location. And so what I ended up with was 773 total paired observations, 430 for corn from 20 different sites, and 343 for soybeans from 18 different sites. And so what I did was I took this data and I developed an economic model to look at the net returns for cover crop systems. And so basically to boil it down, it looks at the cost of implementing cereal rye versus the revenues that you get from the cash crop in those systems. And I looked at two different scenarios, the same as what we looked at for the field scale. And so looking at just the net returns following cereal rye adoption, as well as looking at the scenario where we may receive a governmental cost share payment, in this case represented by EQIP, which is the Environmental Quality Incentives Program. And what I have found is that on average, there's a negative net return from adopting cereal rye into corn and soybean rotations. And so this is regardless of the seeding method that you use, because one is more expensive. Drilling is typically a little bit cheaper than broadcasting, at least for custom rates. And this is also regardless of the tenure of the cover crop system. One interesting thing that I found was that on average, you're 2 times more likely to break even if you drill cereal rye versus if you broadcast. And again, that's related to that cost of the seeding method. And then lastly, as cereal rye tenure increases, so the number of years that cereal rye is planted in the same location, the variability in net returns decreases, meaning that you're not going to see as low of lows and as high of highs. You're going to be more consistent with your net returns. But on average, we still saw negative net returns. And so that's not great, right? But some efforts are being made to mitigate this downside, specifically in terms of vertical coordination efforts. And so this has to do with basically the support of these regenerative practices by firms throughout the supply chain. And I know that within the Ag Econ department here, there's some work being conducted in this area. So all of that was a highlight of the current projects in our lab. And so before I finish, I just want to leave you with the most recent project that we are starting. And that is next generation cover crop management. And so we're calling this precision planted cover crops. And so this is a North Central SARE funded project in collaboration with people at the University of Illinois and Southern Illinois University Carbondale. And it will be conducted across 6 locations, 2 experimental sites, 1 in Indiana, 1 in Illinois, and 4 farmer collaborator sites, 2 in Indiana, 2 in Illinois. And what precision planted cover crops actually is using the RTK guidance on, or GPS guidance on the tractors, basically to plant the cover crops in such a way that we are able to leave an area where the corn row will go bare, so that when we come back and plant our corn in the spring, we have this strip of soil where there's no cover crop growing. And so this leaves us with that non-intersecting zone, which could potentially reduce the amount of nitrogen immobilization that we have to deal with, as far as considering the corn interacting with nitrogen. There's potential that leaving this strip bare could lead to a quicker warm up in the spring of the soil, allowing for better early growth of the corn. There is potential that we could actually reduce the seeding rate that we need to use, because we are being more specific with how we plant. And it gives us some flexibility in the spring as far as termination is concerned. So With cover crops, you typically think that we need to terminate a couple weeks prior to when we would plant the corn. But given that we are leaving a row open where we're going to actually plant the corn, there's potential that we could actually go in without terminating, plant the corn, and then come back later on and actually terminate the cover crop. And so just to kind of get a look at how this practice would play out, we actually implemented it at DPAC this past year. And we had 4 different treatments. And so on the next couple slides, in the top left, it will be what the field looked like at the timing when of cover crop termination. And then the main picture is at corn V6. And so we had a no cover crop control. We had a full coverage, so not precision planted, just normal drilled cover crop at the 100% seeding rate. We had a precision planted cover crop at the 100% seeding rate for cereal rye. And then we had the same precision planted cereal rye, but we reduced the seeding rate by 30%. And so the first thing that we looked at was the performance of the cover crops. And so here is early spring growth of the cover crops. And when we actually sampled the above ground biomass of the cover crops, we found that there was not much difference in the production between any of the treatments. We had between 1,000 and about 1,100 pounds per acre of biomass. And so it was pretty consistent amongst the treatments. And so the next thing that we wanted to look at was just kind of start to get a look at how this might be affecting the actual corn plants. And so some of our preliminary results from when we sampled at V6 show that there was no significant difference in the biomass production of the corn between any of the treatments. And then lastly, we've been working with the farm manager at DPAC, who has been doing some UAV imagery for us throughout the season. And so here is a look at the field from the early dent stage of corn. And you can tell on the NDVI map here that there's really no noticeable differences within the field. And so this field was broken up into quadrants, is how the treatments were put on. And so you can tell between the 4 quadrants, there's not really, without doing any statistical analysis, just visually, there's not really any differences that we can discern. And so this kind of leads us to believe that this practice could be promising, and it makes us excited about the new project that we have going. And so with that, I will be open to any questions if anybody has any.
Richard Jeffalenick, what do you think about the environmental performance of these last set of treatments? Do you think that might suffer a bit by not having quite as full a stand, and how will that be measured?
So that is an interesting question, and yeah, so that is one of actually our main questions: is will we still be able to get the environmental benefit that we saw at that first field scale project by using this precision planted method? And so what I'll say, Dr. Valenick, is that the field that we are doing this at DPAC actually has tile drainage. And then also we're doing this at that same field location that I showed from the very beginning. And so both of those will have tile drainage associated with them. And so what we will be able to tell is if we are losing any of the environmental benefit that we were seeing earlier by augmenting the way that we plant the cover crops. But the other thing that I'll point out is that if you remember from earlier, we're seeing a much greater reduction in nitrate than what is recommended by many of the science assessments. And so as long as, as long as we can maintain competitive levels of nitrate reduction, and possibly see some improvements in the agronomic portion of it, I think that would be acceptable.
Thanks. We look at cover crops, we spend an awful lot of time, and rightfully so, I think, looking at management approaches, different planting rates and dates and ways of killing the rye and whatever. What about designing the rye and the corn in concert so that they are a better match developmentally and physiologically and stuff like that? Or the cover crop in general. I mean, we don't spend a lot of time thinking about idiotypes. What's the best corn hybrid to go into a cover crop situation? And what's the best cover crop structure, morphology, development? Where's that in the conversations in the cover crop world, or are they just not being had?
I can't speak to that very much. What I can say is that, that's an interesting thought. Maybe Dr. Armstrong, do you have any thoughts on that?
Yeah, that's a great question, Jeff. And actually, a few of us that are looking at that in a sense. So what we're doing within my program is that we're definitely looking at these shorter varieties and how competitive they are versus the more mature varieties, so that we can get cover crops on faster. But in addition, we are introducing a greater inclusion rate of overwintering legumes, such as winter pea, balanza clover, certain varieties of crimson clover. And we're looking at these advanced management practices in order to give the farmer more flexibility in the spring when it really counts, when management really counts. And so, for instance, where we have this enviro strip or skipping the corn row, which could allow farmers to do what you call plant green, probably a bit easier. So if you have a legume, like in CPAC, we had balanza clover that put on about 7,000 to 75, about 7,000 to 7,500 pounds per acre of biomass, which generated 200 to 260 kilograms, pounds per acre of nitrogen in this above ground biomass. And then we put nitrogen rates on top of that. And what we're starting to see is that we had zero nitrogen added, and we allowed that balanza clover to kind of grow and mature and fix nitrogen at its maximum potential. Right now, you go to that field, you can't tell the difference between the zero control, where there's no nitrogen, and where we put down 180 pounds per acre. And so we're exploring some of those inclusion of overwintering legumes, different hybrids of corn that could be productive at shorter maturity length, and so forth, in order to kind of advance the agronomics while keeping a pulse on ecosystem services as far as losses of nitrate to the tile drainage.
Thanks.
Okay, does anybody else have anything? Okay. If nobody else has questions, there is one thing that I would like to share with the group before you go, especially since there's quite a few faculty members in here. And that is, can you all see my screen now?
Yes, we can.
Okay, so this is a program that, and I'm not going to take credit for this. This is so Adabacola Dada, Charlotte Lee, Christina Schilling. They've really, and Dr. Boyne has been part of it as well. They've really been the champions of this. But this is a program that we, as a graduate student organization within the Department of Agronomy, coupled with the Office of Multicultural Programs, has put together for all graduate students within the College of Agriculture. And so we feel that this is a very important thing that we're doing. We're very proud to be at the leading edge of this within the college as far as programming for graduate students in this area. And so my request to the faculty and that are in here right now are to please encourage your students to participate in this. If there's any questions from anybody right now, Charlotte and Etibacola, I think, are both on the line. And so they may be, and Dr. Bowling is here as well. And so they can help me hopefully answer them. But I just wanted to show this now. It will be spread around. You will see official announcement coming early next week via email. But we wanted to share it with all of you today. So,
Richard, is this just within the department, or is the intent to go beyond agronomy?
No, so this program is open to all graduate students in the College of Agriculture. So it is not just agronomy. It is for, animal science, ABE, everybody, right? So, and that was important for us because, it's not just agronomy that needs it. So
No, I agree. What now is this going to be a Zoom type thing or WebEx or what? It doesn't say. It says register, but what is it? What's the format?
yeah, so it is Zoom. It is via Zoom, and the registration is essentially so that we can make sure that who's supposed to be in there isn't. You know what I mean? So it's only students. And so that's part of why there's the registration. And so once you register, the Zoom information will be sent to you.
And I'm going to add on a little bit. Is that okay, Richard?
Absolutely.
This is Charlotte talking. And so this is kind of the first event in a series of workshops. So the idea is that throughout the year, there will hopefully be multiple opportunities to engage. Not necessarily monthly, but maybe once or twice in a semester, there will be an event that students can engage in. And this is the first one. And so in this event, we're hoping to address kind of the historical framework of racism and how racism is manifesting itself in the agricultural system and kind of local realities to us in Tippecanoe County and surrounding areas. So it's really more of an information kind of background. And it will be followed by an opportunity for individuals to discuss kind of their experiences and racial identity, building upon this knowledge. So those will be small group or paired discussions. And so that's another part of the registration, is that's going to help with the pairing of the students in the Zoom breakout rooms. So it's a combination workshop where there's information provided, but we also want to work on having open communication and conversations among our peers so that we can, move forward in creating a better environment for graduate students.
Well, thank you. It's really important. Thank you for taking the lead on this. I mean, it's critical. Longer term, this is a great first step. Longer term, are we going to develop programming that's broader beyond just race to include other marginalized groups?
Charlotte.
That's a good question. I think that's something that we can definitely keep an open mind about in our upcoming workshops, and developing and thinking about this as officers of the graduate committee. I think all of those things are definitely important to us, and this is just kind of a starting point. And we are hoping too that this curriculum that we've been working with the Office of Multicultural Programs to develop is going to be something that could potentially be offered, each year. There may be slight adjustments to it, but we do definitely have concerns for other marginalized groups as well, and hopefully we can include that into future programming.
Is there a coordination among other land-grant universities and things? This isn't unique to Purdue. The reason I raise this is because the American Society of Agronomy is identified as one of its 3 strategic initiatives for the next 3 years as diversity, inclusion, equity, and justice. And so this will be going out to, that plan was just approved, and it'll be going out to all of the institutions, and there's a lot of interest, I know, nationally in this. And I'm just wondering if there's any coordination or any thought about coordinating it, because I think it's a huge step forward. This is really, really super.
I think ideally we would like there to be some more coordination. We first started planning this way back in May or June. So this is something that has taken a long time to pull together. And one of the reasons we were pursuing it at that time was because we know that sometimes it takes a while to get the ball rolling. And so, it may be a while before we see something that is, required or coming down from a college level or university level. So we wanted to start with something we could do here in our department, but also have it open and hopefully use that to kind of gain momentum for change. So that's kind of where we were coming from originally on this. Was it was something that was brought up by students in our department as something they thought that, we as a graduate student organization can make this change, and hopefully, it's difficult to say something is required, but working with Dr. Bowling and hopefully the graduate committee, maybe we can have something where participation in these events is weighed upon in terms of thinking about where awards or scholarships are given. Exact details still working out, but we really think that this is something that's important, and we would really like to see participation from all graduate students in the department. We know it's difficult to coordinate everyone at one time. Ideally, this would have been kind of built into maybe an orientation. Maybe that's something we can do in the future, but we're just kind of, like you said, this is something that's important, and we're developing, so we'll make sure to keep everyone in communication about what's going on.
Well, I think grassroots efforts are often more successful than top-down things. So this is wonderful. I mean, coming up from those that care, those that are committed, rather than being told by a department head, a dean or something, so this is great. So if I can help in any way, being involved with the Tri-Societies to extend it beyond Purdue and Purdue Agronomy, just let us know. There's keen interest out there. And I'd love nothing more than Purdue to be at the cutting edge, the Purdue grad student group to be at the cutting edge of helping work on this situation. So thank you.
Thank you. Okay, if there is no more questions or comments, we've been on here for an hour. I don't want to take any more of anybody's time, but if anybody does have anything, we can address it now. If not, this, like I said, this announcement will be sent out via email on very soon, and you will be receiving it, and as well as all the students. And so please, like I said, just encourage your students to participate. And with that, I just want to thank everybody again for participating today, and have a good weekend.
Thanks so much. Nice job.
Thank you. Good weekend.
Thanks, Richard. I'm signing off.
Hey, Charlotte, sorry I threw that on you.
Oh no, that's fine. I'm here to help support and answer questions.
Yeah,
So-
you had been more involved, so I just wanted to let you handle it. So
I appreciate that, and great job